One of the Con Toolbox's best features is its graphical user interface. Virtually everything you need to do can be done from the GUI, and Con's layout is straightforward and easy to use, especially for newcomers. The first step of any Con analysis is to create a new project. Clicking on the New button will prompt you to enter a new project name. Let's call it Arithmetic. As you make changes to the project during pre-processing and certain analyses, you can overwrite the project file at any time by hovering your cursor over the project menu and selecting Save. You also can close the Con GUI if you need to, reopen it at a later time, and load your project by clicking the Open button and selecting the project file that you created. As you use the Con Toolbox more, you will encounter errors that are specific to your analysis. The Help menu at the top of the window contains links to the Con manual and other web resources. The Search Help option opens a search menu to filter forum posts by topic. For example, if you want to see every post that mentioned global signal regression, you can just enter it into the filter field and press Enter. In order to make the GUI easier to read, some users may want to change its appearance. Hover your mouse over the Tools menu and select GUI Options. This will open a window that allows you to do things like increase the font size or change the color scheme. For example, we could change the background from black to red. This looks hideous, so I'm going to change it back, but you get the point. You will notice that along the top of the Con GUI, there are 4 tabs. The Setup tab is where the user enters the scanning parameters for the study, such as the number of runs and the TR. There are also buttons for loading the structural and functional images. In this experiment, there was 1 resting state scan and 1 anatomical scan per subject. Since we are beginning by analyzing just 1 subject, we enter 1 for the number of subjects, and also the number 1 for the number of sessions or runs field. From the JSON file on the OpenNeuro data download page, we learned that the repetition time, in other words the TR, was 3.56 seconds. The acquisition type menu provides 2 choices, continuous and sparse. Most experiments will use the continuous acquisition. Sparse acquisition is used for event-related designs and omits convolving the HRF with the onset of each trial. For now, leave it as the default of continuous. We now move down the left side of the GUI to the Structure tab. Click on it and then use the menu on the right-hand side of the GUI to select the file sub01 anat sub01 t1w. A pop-up window will say that 1 file has been assigned to 1 subject. Click OK, and the structural image will be loaded in the center window. The slider to the right of the image can be used to flip through different slices. And the plus button at the top of the slider will change the viewing montage between axial, coronal, and sagittal slices. Clicking directly on the slices will open up another display window that allows you to look at the slices in combinations of planes by clicking multiple checkboxes. The Functional tab is similar to the Structural tab. In this case, on the right-hand side of the GUI, you can use the navigator to select the file sub01 func sub01 task rest bold.nii, and it will load the image's slices into the center window. The first volume in the time series is displayed on the left, and the last volume in the time series is displayed on the right. If there was any major movement or artifacts between the beginning and the end of the time series, it would show up in this side-by-side comparison. If there was little or no motion, on the other hand, the 2 images should look virtually identical. As with the structural tab, you can switch between viewing planes and flip between different slices of the functional data. There is a difference, however. Instead of opening up a new viewing window, clicking on the slices themselves will open up a time series plot, extracted from the voxel that you click on. Since these data haven't been pre-processed yet, you may notice trends in the direction of the time series, either upwards or downwards. These represent scanner drift artifacts, which we will later correct during pre-processing. Another useful check to do at this stage is to click on the Functional Tools menu in the bottom left corner of the functional data window and select Slice Viewer with Anatomical Overlay, QA_reg. This displays the functional data and anatomical image simultaneously and traces out the major sulcal and gyral curves in yellow. Check to make sure that the boundaries of the gyri and sulci of the functional data roughly match up with those of the anatomical image. Selecting both the coronal and axial planes is a good way to check that the ventricles and other internal structures are aligned. If you like, you can do the same QA check with the SPM CheckReg button by clicking the Functional Tools menu and selecting Display Functional Anatomical Co-Registration SPM. This will open the CheckReg window, similar to what you used in the SPM tutorial. There are many other options in the Functional Tools menu. As an exercise, take some time to look at each of them and think about what they do. You may notice that there are other options in the GUI, such as ROIs, conditions, and covariates. We will leave those for now, coming back to them after we have pre-processed our data, which we turn to in the next chapter. Pre-processing a resting state dataset is similar to how you pre-process a task-related dataset. You do the same steps of realignment, segmentation, normalization, and so on. The main difference is in the motion threshold. Resting state datasets are notoriously susceptible to motion-related artifacts. Even small movements can introduce spurious correlations. These artifacts are particularly problematic for laboratories studying disorders such as schizophrenia, or for researchers who scan very old or very young people. Too much movement will ruin your study. There's no simple way to address this problem, aside from training the subjects to move as little as possible. That said, the Conn Toolbox has several methods for mitigating movement artifacts, including ART and principal component filtering of signal from the white matter and cerebral spinal fluid, a method known as CompCore. In the following chapters, we will examine how including or omitting these steps will affect our results. Our first step is to pre-process the data to improve the quality of the images and reduce noise. To perform all of the conventional pre-processing steps, as well as removal of movement artifacts, click on the pre-processing button at the bottom left of the screen. This will open a menu showing virtually every pre-processing step available. Bold font indicates a pipeline, or series of pre-processing steps that have already been arranged in a particular order. The first option that is highlighted, Default Pre-Processing Pipeline, will do a traditional analysis that normalizes volumetric data to MNI space. Clicking OK will add all of the pre-processing steps into the window at the top of the screen. Highlighting a step will display a short description of what that step does, along with the input and output. Some steps, such as functional direct segmentation and normalization, allow you to specify additional options, such as whether you want to use the first functional volume as reference. These options probably won't affect your results much, if at all, but they are there at your disposal. You can also move certain steps up or down in the pipeline, or add and remove certain steps by using the Add and Remove buttons on the right side of the menu. For now, however, we will click the Start button to begin pre-processing the individual subject that we have loaded. Before the pre-processing can start, however, you will be prompted to enter a few more options. For example, you need to specify the slice order, which should be on your scan log. If you're unable to locate it, ask your scan technician to find out what order was used. In this tutorial, we will use the interleaved Siemens ordering. You will also be asked to specify the threshold for how ART identifies an outlier. The intermediate settings, 97th percentile, should work fine for most cases, although you may want to set it to a higher or lower threshold depending on the population you are studying. Instead of basing the threshold on a normative sample, moreover, in other words, the settings that have already been selected, you can instead choose to edit the settings directly by selecting Edit Settings and manually selecting the subject motion threshold. For our current tutorial, we will leave it at the intermediate settings. You will next be prompted to select the sampling resolution of the anatomical and functional output. The defaults of 1 mm cubed for the anatomical image and 2 mm cubed for the functional images should be fine. If you want to take up less space on your hard drive, you can lower the resolution, in other words, increase the numbers in the fields at the expense of lower spatial resolution. Finally, you will be asked to specify a smoothing kernel. As you will see later, the smoothed data by default is omitted from the actual functional connectivity analysis. It is included here in case you want to use it. Click OK and the pre-processing will begin, calling upon SPM tools as needed. For this subject, it will take about 5 minutes total. If everything has run without error, you should see a pop-up window saying that everything has finished without any problems. Now we will need to inspect the images for any artifacts or other problems. In other words, we will do quality assurance or QA checks. When pre-processing the data, a good habit is to examine the images before and after each step to determine whether everything ran without any errors or if instead there was something you need to double check or redo. We will first use the buttons on the left side of the Con GUI to examine the output of some pre-processing steps, and then use the QA plots button in the bottom left of the Con GUI to create a comprehensive QA report. Once the pre-processing is finished, click on the Structural tab again. You will notice that the anatomical image looks different. In fact, it has been skull stripped, resampled to 1 mm cubic resolution, and normalized to match the MNI template. Scroll through the different views to make sure that the brain looks as though it has been normalized appropriately. We can examine the image more in depth by using some of the viewer options. If you click on the Structural Tools menu and select Slice Viewer with MNI Boundaries, QA norm, you will see the outlines of the MNI template traced in yellow on top of the anatomical image. Observe how the yellow lines roughly outline the boundary between the white matter and the gray matter. The normalization won't be perfect, but the general outlines should match up. Clicking on the Functional tab reveals a similar change. The functional data have now been fully pre-processed, which includes normalization and smoothing. Clicking on the Functional Tools menu and selecting the Slice Viewer with MNI Boundaries, as with the anatomical image, will show a yellow line dividing the gray and the white matter. The general outline of the MNI template should also encompass the functional image. If both the anatomical and the functional data are aligned with the MNI template, this indicates that the pre-processing was probably successful. We now come to the Regions of Interest tab, which lists the different regions that will be used in the connectivity analysis. 5 sets of ROIs have already been generated for you: gray matter, white matter, cerebrospinal fluid, atlas, and networks. Let's begin with the first three, which correspond to the tissue types that make up the brain. Clicking on any of these slices will open up a montage window that you can use to more closely examine the overlap between the gray matter mask and the gray matter of the functional image. While you're at it, look at the white matter and CSF masks as well to make sure that they seem to have been segmented appropriately. The next output is atlases, or ways to parcellate the gray matter into different nodes. By default, the gray matter is partitioned according to the Harvard-Oxford cortical atlas, and the cerebellum is partitioned according to the AAL atlas. In order to see these atlases in more detail, clicking on the parcellated 3D brain will open a viewing window illustrating how the atlas has been resampled to the functional data. For example, the parcellation colored in orange indicates the anterior cingulate. Hovering your mouse over the different colors and clicking will show which ROI you are pointing to. Later on, we will use these ROIs in our first level analysis to generate connectivity maps between those ROIs and all of the other voxels in the brain. The same idea is illustrated in the Networks tab, which assigns groups of nodes to a single network. The default mode network, for example, is a combination of a node in the posterior cingulate cortex and a node in the ventromedial prefrontal cortex. As with the cortical ROIs, you can open up a new viewer by clicking anywhere on any of the slices. If we click on, say, the blue area, this is one of the nodes of the default mode network, the posterior cingulate part of it. If we look at other slices, we can find other parts of the default mode network as well, for example, within the medial prefrontal cortex. Another output from preprocessing is nuisance regressors, or covariates such as motion that we want to disentangle from the signal we are interested in. Clicking on covariates, first level, reveals three covariates that have been generated by default: realignment, QC time series, and scrubbing. The realignment covariate is a set of 6 movement parameters representing movement in the 3 translational and rotational directions. Hovering the mouse around the movement parameters will display a vector of 6 numbers representing the average motion in all 6 directions at that time point. The second covariate, QC time series, computes an additional measure of motion called frame-wise displacement, or FD, a type of composite measurement of all of the movement parameters. The default in Con is the FD computed by the ART toolbox. If you want to use another FD formula, click on Covariate Tools and select Compute New Derived First Level Covariates, and you can select from any of these options. The last covariate, scrubbing, will display any volumes that have been modeled out of the data, or scrubbed. This subject was very still during scanning, so no volumes have been removed. Later on, we will see subjects that have had volumes removed by scrubbing. You can summarize all of the QA checks that we did above by clicking on the QA plots option in the bottom left corner of the Con GUI. In the window that opens up, click Create New Report and label it whatever you want. Then click Create New Plot and select any of the QA checks that you want to include in your report. The default ones that are highlighted will display checks such as the functional and structural data on the MNI template and the motion parameters. When you have chosen all of the QA plots that you want, click Start. A series of figures will be generated, one for each QA check that you selected. You can then click on the Export button to generate an HTML file of all of your QA checks. When it is finished, you will see something like this that you can open up in your web browser and click on the links to review any of the pre-processing steps. If you are satisfied with your QA checks, you are now ready to begin denoising. This will further clean up the data using processing specific to resting state data and prepared for statistical analysis.